One of the subjects of the overall Combine meeting, which is how do we make models talk to each other? And in my case, I'm interested in model validation, so guaranteeing that the underlying biology is correct. So this is work that I did during my PhD. And during that, I had two fronts of tackling the issue. One was building a translator to go from PhysiCell to Computational, and another was building a Python package for cell behavior that I won't talk about. So what we want to do with ABMs is to go from the top picture to this somehow. And how we do that depends on what we're trying to do and what we care about in our model. So lots of differences. But there still are a few things that we should be able to agree on, which are things like, what are cells? Are they alive? Are they moving? And also, what is the physics of the issue, right? Cells are in a very viscous system, so we are not in a Newtonian situation. And because we are in a computer, we have to define everything. We don't get anything for free, as James likes to say. So as I said, there are a myriad of different methods, fine-tuned for what, a specific problem sometimes. But since we are trying to represent reality, we should be able to go from one method to the next with some information loss and whatnot, but should be doable. Yeah, I can just stand here.
Yeah, okay.
So what's the issue right now? ABMs are not fair. They are not, well, they are findable and accessible, but they are definitely not interoperable. And most of the times they are not reusable. So Chichi experienced this by having to re-implement the TB model to find new stuff. And that's something that I think a lot of us have done in the past. So it would be nice to not have to reinvent the wheel a lot of the time and actually just make a better wheel instead of starting from zero. And besides that, this issue also inhibits collaboration because, well, I want to use my method, not yours. So let me remodel everything, or just, well, I'm just going to do my thing. So is there a way to change that interoperability? One way, which I've been advocating, is to have a universal model spec for ABMs. Then each platform would be responsible for reading and interpreting, which is a big dark forest with many unknowns, and it's a hard problem. And there are a few things that I believe about this universal spec, like that we should prioritize defining objects like cells, because a cell is going to be a cell everywhere, instead of how to make the cell move. And this universal spec needs to describe many things, like metadata, what are the containers, the environment the cell is in, the cell is a container for other things, dynamics, events, and so on. And so, of course, that's very big and very hard. So I asked myself if there was something that I could do as an intermediary step to find some of the problems and hopefully find some of the solutions. So what I did was to build a translator to go from PhysiCell to Computational. And something that was mentioned during Combine so far was comparing Morpheus and Computational, which are both cellular POTS model. So it should be easier than this. But in my opinion, because it is easier, it wouldn't be as informative. If it is doable to go from Morpheus to Computational, great, obviously. If it wasn't possible, I would vote for that, saying something more about Morpheus and Computational than the idea of a translator. So going from PhysiCell to Computational is harder because PhysiCell is a center-based model, while Computational is a POTS model. So what the translator has to do is to read the original specification for PhysiCell, interpret it, reorganize in a way that CompuCell understands, find solution for missing concepts and other issues, let the user know of any problems, let their user know about things that they should pay attention to after the translation is done, and then write everything out to files. And the main problem with a translator approach is that this is already out of date because PhysiCell had some updates that changed the configuration files. But anyway, so a few more differences of PhysiCell and CompuCell. PhysiCell uses XML and C++ to define the models, and CompuCell uses XML and Python. And one of the limitations of the translator is that I will not translate C++ code. I have no idea how the coder did that. I have no way to make a universal interpretation of that. Few other important differences is that cells in PhysiCell are center-based and they don't have a shape, while in CompuCell they do have a shape and are made of pixels, so sort of like a picture of cells in vitro. And PhysiCell is force-based, CompuCell is energy-based. Yeah, and PhysiCell is off lattice for the cells, but on lattice for chemical fields. A bit more, just an overview. PhysiCell dynamics, you have a sum of forces that will define the velocity of the cell. Remember, we are dealing with Newton, so this is fine. And so forces drive velocity, while in CompuCell, the energy changes will result in the velocity. So it's a bit different in my mind, at least. And also in PhysiCell, it's a global sum of vectors over the cell, its neighbors, and everything else. And in CompuCell, the calculation is always done at the border of the cells. So it's a local process. So in short, what the translator does, it parses the XML from PhysiCell, just the settings XML file, interprets that data, and it translates by itself the cell types and their constraints. So things like what volumes should the cell have? Does it do chemotaxis? Does it secrete or uptake from any chemical field? What phenotypes does it show, which is the other Python package that I won't get into maybe this afternoon. Also, what are the fields there? Oxygen, glucose, whatever. What is the field's diffusion and decay rate, and so on. How the cell should do secretion and uptake of chemicals. What are the boundary conditions? And I think that's pretty much it. What it doesn't translate yet is the initial cell configuration, and I'll talk about why in a minute. How to switch cell behavior, and of course, any hand-coded instructions that are in C++. So does it work? I have two examples. There we go. This is cell cycle. The simulations run in slightly different time steps. So one thing that the translation does translate is what should the time step be in minutes. And also what should the pixel or voxel size be in micrometers or whatever. But the two simulations end up running at, or my GIFs run at different speeds. So this is cell cycle. The cell is changing behaviors, growing volume, and dividing in both situations. And the things that I had to do after hitting the translate button was to define the initial cell position and create this color map. And I also increased the cell volume to make it easier to see on the right.
So here,
yep.
The color map that are like cell states.
Yes, the color maps are the different phases of G0, G1, S, G2, and F. Yeah. Yep.
What about cell shape? You said that.
Yeah, PhysiCell doesn't have any cell shape. They are just spheres.
So what do you do?
I tell CompuCell that they should have a volume.
So they're circular?
They tend to be circular because of contact surface energy minimization.
Because these cells have a variable volume.
Yes, they have a variable volume. Yeah, they actually are growing here, but it's hard to see.
I guess what I'm getting at is because these are more challenging, different frameworks, any translation is going to necessarily be lost.
Yes.
So that's what I'm really asking. What do you do with that lost information?
For now, I'm living with it. But yeah, that's a big issue. I try to minimize the loss, right? And in this case of the volume, I'm actually gaining information, right? I didn't have any information about shape, and now I'm getting at, yeah. But I had to make it up as well. Yeah. Yeah. What was really lost were more things to do with. So in PhysiCell, you can say a cell is going to move at this speed, and it will do that. That's something that I don't know how to do in Computational. Yeah.
Well, I mean, some of this problem is sort of model specific.
Yep.
And that, I mean, some with models that you impose to POTS or center model, and some is due to differences in methods. So center model approaches will model implicit cell shape by treating cells as being something like spherical or round, in which case you impose interparticle interactions according to equilibrium distances. So my cell has a radius, therefore it has in 2D an implied area or volume. The thing is, when moving into POTS, depending on how you're modeling cell shape, there are potentially multiple equally valid configurations for cell shape. When you say my cell has an equilibrium area,
yep,
you can assign probabilities of finding a cell in a particular shape that has that area. And according to POTS, all of those are equally likely.
Yep. And you can kind of see that here. It's kind of fast, but you see how they have different shapes all the time, but they keep the volume constant-ish when they are not growing.
I'd like to point out that you're not actually losing information because you have the original simulation also. You are gaining information.
Yeah.
When you do the translation, don't throw away the original.
Yeah, definitely don't throw away the original. The idea is to compare the two and see if they agree, hopefully, or disagree, and then you have a problem.
So having written the translator, if you imagine that there is a universal intermediary between those two things, for any one model, would you imagine that model being based in a center-based world, and that if you were to translate it to Computational 3D, that becomes a new model? Or would you imagine encoding all the information into a single package and saying this is the model, and it has some information that is center-based specific and some information that is Computational, that is POTS specific?
I see it as having a model definition file that is as agnostic as possible, method agnostic.
Sure, but you need this. But if you want to change it from exchange between POTS models, simulators, you would want all that information in there.
Yes.
So would you put that into the model, even if, would you imagine that being the same model as something that has a single model with all the information, everything you would need to run it on any platform? Or once you do that, are they sort of I don't know, appendix-like, and they don't have any function, and they're likely to get out of sync with the rest of the model or something? Would you want just one as sort of your default? This model is as translatable as I can, but it is made for POTS.
Right.
And then if I change it to a center-based model, I go ahead and drop the POTS things, I add the new things that I need for the new one, and say this is a new model that's based on this one, but it is,
So I think when you make the model description file, which is the universal spec, you put the information you need there, but try to avoid putting information that is specific to a method, maybe, if that's possible. I'm not sure it is.
Well, if you
and then
put it in, then you've got
recreate it from scratch if you move from one plots to a different model. Like if you move from Computational 3D to Morpheus, then you could have saved information that Morpheus could have used, even if PhysCell couldn't. Right?
Yeah, but I'm not so sure how much of that information is needed or exists to rebuild the model, right? If I have a specification that I say I have the simulation, it's 200 microns by 200 microns, and I'm going to start with 5 cells in there that are epithelial. And I define some rules for behaviors they do in different circumstances, and that's it. Then you read that and you have some software that reads that and spits out some standard beginning, at least, for a model in whatever.
The one thing I remember is like how you calculate order distance.
It's different in Computational and Morpheus.
Or Morpheus, right? Was different, and that seems like an important thing to save in the model if you wanted to copy
Yes.
them before, but it would be useful for PhysCell.
Yeah, that's very true.
At least now.
Maybe that's something to say with the data with the data outputs of the model instead of the model specification.
The model in the model itself.
No, you would save it. I mean, I would save it in a separate CSV, but I would note somewhere in that CSV metadata that, hey, this perimeter was calculated using this.
way. So you imagine like a separate file that contains a more detailed information about the model for a particular platform.
Yeah, maybe.
I think this gets at changing levels, right?
Yeah.
So
Mhm.
at the basic biology level, we want to say this is the same model. It's about this stuff, right? Yeah. But then I think that you want some naming scheme that captures, okay, now here's specification of the model that will leverage this kind of mathematics.
Yeah.
Versus here's a different specification that will leverage this other kind of mathematics. They're both the same biology, but we better name them different because they're going to be used in different simulation environments.
Yep.
We are getting here into prominence, basically. That when you change a model, then translation is the same change of the model. They need to record what you do and what you drop and what you add. And these can be done either separately or within output file, which basically is the same, just like add RTF to the code as annotation. But yeah, you need to have certain steps to record your actions.
Yeah. And going a bit back to, do I see them as being the same model or different models? I see them as being different interpretations of the same script.
And would you see it as one file or different files?
I don't know. Yeah, but skipping to my final slide. This is a mock universal thing that I made in 1 hour. So it has some of the things that I'm thinking that should be there, certainly not all of them, but I think this should be sufficient as a mock first thing that can be read by Computational, Morpheus, PhysCell, and whatever else. So I just have some units and saying that the simulation is 2D, the size of the simulation, some more units and how long the simulation should be in that unit. And here I have a epithelial cell. It has a volume, a proliferation rate. This migration speed might be problematic, but it's there. And a death rate and a simple behavior. So if it is below a certain neutron level, it's going to try to find a higher level. How you're going to do that, up to the modeling framework. If it's reached that, it's going to divide on some initial setup. How much time do I have left?
Yes, but you can say a few words more.
Yeah, well, I was going to talk about issues that I found, but I can do that this afternoon during my session. Questions?
Yeah. Is that all questions were asked during the discussion?
We never heard of constant-ish before.
But I think it's a pretty good way of describing it.
A question about this.
Yeah.
What role does this play in the rest of what you showed. Because you have a translation from
Yes.
PhenoCell to CompuCell. I think all of this would come along in that translation.
Right, yes. So, but in my introduction, I was saying that I would love a universal model spec, but I can't do that. So I'm doing the translation to find some of the issues.
So this is like a different route.
Yes.
This loaded.
This is the, not even the first step of the different route.
Is this actually all the information you needed for that particular translation?
No.
Okay.
This was something else that I
Okay.
just, yeah, it's a mock.
Question about the specification of behavior. I mean, that would be the bit that would generate the C++ code on one side and the Python
Yes.
on the other side.
Yep. That would be converted to C++, Python, whatever.
You would envision that a controlled language there.
Yeah. So because we need some flexibility, I wanted to avoid XML, right? So
But I mean, you would have to know about gradients moving towards something.
yes, yeah. This is implying that you know gradients and that you have some way to sample fields and that you have fields. So if you don't have, no, that is true. I don't have a definition of nutrient level.
Is that something you imagine that it would be a universal?
Well, I mean, for initialization, you have it, so it would have to be appearing somewhere.
Yeah, and I mean, I'm telling the simulations to sample nutrient level, so I should have some agent type field.
Are you imagining that nutrient level is a model specific thing, or are you imagining nutrient level as a universal all?
Well, in this case, I was thinking of nutrient level as glucose diffusing somewhere.
Okay.
So how you represent that is going to depend on the model, but it's usually, yeah, it's going to live in the environment.
I mean, that's for the sake of, because you will have a session.
Yes.
Okay, yeah, so that's the.
So just quickly, in addition to visually comparing them, do you have any quantitative metrics for saying how, how translatable whether
Right. So I don't have it in this presentation. I have in some PowerPoint somewhere. I compared for this one, I compared the population change. And it
Number of
was,
cells.
yeah, number of cells. That was pretty close. And this other one, which is going to be the subject of this afternoon talk. So the cells are picking up cargo and delivering cargo. I didn't actually have any metric for this. I couldn't come up with one. On the left is PhysiCell, and on the right is CompuCell. In the middle, it's CompuCell before the extra translation steps that I had to take.